Hey guys, welcome to Unknowns today. Today's presentation will be on how to identify a fungi. Okay, so fungi, it's a general word. We have a three entire group of organisms called the molds, yeast, and mushrooms based on their how they grow. Okay, so I would say personally that yeah, basic quality of mycologist is that those who study fungi is that the ability to produce a clean control plate. When you use a Polydextrose agar or Subculture, your plate should be clean, meaning you should not contaminate with the environmental fungi. The second thing is to producing and maintaining pure cultures. Again, it's just a repetition of the first condition. If you are able to produce a clean control, you will be producing a pure culture. That is a culture with only one species of fungi. The third thing will be producing a countable fungal colonies. If I give a water sample or soil sample, you should know how to serial it and in what dilution you will be getting a countable numbers, not in a form of a MAC group. So today's presentation, I would say I will talk about identifying fungi from soil, from leaf surface and endophytes, that is within leaves. Okay. Before getting into lab, you should remember these three things. Okay. First thing, you should visualize your experiment. What are you going to do? Second thing, execute it in very sharp manner in very limited time. Do not waste your time while doing experiment. Third thing, just clean the lab and leave it as a better place for next people who are using the lab. So while concentrating on different experiments today, I will be talking about how to document the fungal morphology because it is very much important. The documentation to start from your source of isolation, from where you are going to isolate your fungi. Even the source of isolation should be documented. Okay, so what are the fungal pictures I'm going to show in this presentation? Basically isolated from these sources. All right. So fungal identification will be of three types: macromorphological, micromorphological, and molecular. Of course, if you want to know the species name, molecular characterization is the only way, especially DNA sequencing is must for identifying a species, any microbial species in that sense. First thing is fungal colony characterization. In macromorphological characteristics, you need to know how the fungal colony looks like. Yes, you need to document the reverse plate because reverse plate produces venation, which is very much essential to identify the fungi. Spore color, that could be a black, green, or you can see the colors. And there will be pigment production. Some fungi will produce pigment just usually in the reverse plate, which is very much evident. Some pigments can be diffused in the media. So mycelial structure in micro, while we do the lactophenol cotton blue staining, we will go for mycelial structure, hyphal structures, pore bearing structure. All these structures has to be observed microscopically after lactophenol cotton blue staining. Finally, ITS gene that is being proposed as a universal DNA barcode for fungi. Few uses protein profiling also. Okay. So how to document and see whenever you work with water, soil, or air samples, this is how the mother plate looks. This is a mixed culture, meaning it contains more species. You need to isolate, you need to sample one species out of it and go for pure culture. That is, all the plates I'm showing here usually potato dextrose agar. You have to inoculate at the center. What I'm showing is day one culture, how they gradually grow, day three, day four. You need to measure the size of the colony every day. You need to document like this, the front picture and the reverse plate picture. These are the corresponding reverse plate pictures of the front pictures. So this is how the macromorphological features should be documented carefully. The spore color and how they grow, every day these pictures has to be maintained. Okay? Pictures should be this clear and this clean. So when you compare your picture with the microcues, a software available for identifying the form molds and other manuals, your picture should be very clear because you should not take a picture like this, okay, where the cameras are visible, the photo, the reflections and everything is there because this render your actual macromorphology unclear. So nothing should be abstracting the picture because each and every fungi will have their unique macro morphology in front picture and also in reverse picture in a specified media If you change the media, obviously just the morphology, you can expect them to differ. Here is an example. See, Penicillium expansum, they grow differently in different media. The morphological character of front and reverse picture will be changed. So this is the morphological character. Micromorphology, this is where we are looking after lactophenol cotton blue, phenol which kills the fungi actually. Lactic acid in the lactone, they preserve the fungal subcellular structures. Actually, the cotton blue is the one that stains the cellulose component of the fungal cell wall, which render the fungi to look like a blue color in microscopic analysis. So the hyphae can be a septate as shown here or aseptate hyphae, but it should be very clearly seen, not like this. Okay. Usually, I recommend you to go start with 40x and end up in 100x. While you end up in 100x, try to focus on the individual spore bearing structures. These are very much important characters for identifying fungal species. This is how a penicillium usually looks, aspergillus, nucleus, you may not have any information from here. So the picture should be very clean. Even the spore morphology, okay, it should be very clean. It should be oval or spherical. Just by looking the spores, we should be able to see. Another important interesting character of fungi is they produce a unique morphology in the broth cultures. That can be a pellet-like morphology, or you will see mycelia growing above the surface of the broth. All these characters are very much important for identifying the fungi. So meticulous documentation of the character is very much important. So once you have this is how from if you have done the dilution right, you will have isolated colony. This is something that has been isolated from soil. Here I have a protocol for isolating an endophyte where basically you will surface sterilize the leaf, okay, using different surface sterilants like sodium hypochlorite, ethanol, and rehydrate the leaf in the distilled water. You will imprint the leaf on a potato dextrose agar and inoculate in the different plate. Imprinted plate should not have any growth, meaning the surface sterilization work, nothing, whatever fungi that has been present as an epiphyte or on the surface has been removed. The inoculated plate should have a fungi coming out of the leaf biomass, indicating it as an endophyte. Yes, many plants have fungal endophytes within them. And when you change the protocol, I have seen in my lab when we change the even timing, instead of 5 minutes, if you treat them for 3 minutes, if you change the timing for the ethanol treatment, you will have a different fungi growing. Yeah. The diversity of fungi cannot be all diversity or all the species composition within the leaf cannot be isolated in a single stroke. We have to optimize the protocol for every plant species that we are using for isolation. This is how usually endophytes will look. We usually this is the reverse plate picture where the inoculation is done by stabbing the agar from the place of plant inoculated. You will see fungi coming out. See how to know they are endophyte or not. You see, when I put this leaf in the microscope, I can see molds coming out of the leaf surface, filaments of the hyphae that is coming out of the leaf surface in 40x, and this is how they look. Okay, so we should be able to distinguish an endophyte or not. See, this is a bacterial contamination. It is very clear here. It's not an endophyte. This is a fungi or mold that is coming out of the plaque. It is an endophyte. All right. So maximum of 4 inoculation of plant samples after treating with the surface sterilant can be used in a single plate. You will have plate something like this. Okay, see, even though same species here has been inoculated, but we have different fungi coming out of the leaf. You will also have a bacteria or yeast coming out of it in many times. So you may have surprises also like this. Sometimes you will see a mushroom coming out of it. Sometimes, the imprint plate is very clean after ethanol treatment or hypochlorite treatment for this leaf. But I have seen a mushroom coming out of their plant leaf. So finally, sometimes or in many times for endophytes, you cannot, see a proper sporulating structure for which you have to go for a slide culture technique. Yeah, outline picture has been shown here. Probably next video I will talk more about how to do slide culture. But this is the basic difference we will see. This is a fungi that is being teased out from a PDA plate and done with lactophenol. A fungi that has been trained after a slide culture. See the details, the maximum amount of details we can capture in slide culture. So finally, the easiest method of preserving fungi is just make agar buttons of a well-grown plate which is covered with the molds. Make an agar button like this and put it in a 5 ml tube and fill it with that sterile distilled water. You can use preserve like this in the room temperature for years to come. More than 10 years it can be kept like this. So after that, after preserving, if you just pick a agar button and just put it in the center of the PDF, you will see the entire fungi coming. So to identify a fungi, this is the protocol or the flow through I will suggest. With the traditional taxonomy, you will end up in knowing what are the macro and micromorphological features you can see. You have to go for molecular taxonomy. Usually, I will isolate DNA and we will amplify for ITS gene sequences. Then we will send it for sequencing. Usually, the cutoff range of more than 99% similarity usually indicates same species. So this is the overall flow through for identifying a mold species, especially. Um, yeah, if you have any doubt, please post it in the comment. I will be answering you. Thank you for watching the unknowns.